Okay, today we're talking about cellular respiration and fermentation. We're going to be using oxygen and carbon dioxide sensors to measure concentrations of these gases. We'll learn about aerobic and anaerobic cellular respiration. And this lecture is mostly going to be about describing the inputs and outputs of aerobic respiration and fermentation. Whee! I am using slides from the Pearson book, which you may not be able to see the title down at the bottom. At any rate, fancy pictures, gotta love it. So, ATP is our friend. That is a molecule that stores energy in this bond here between the second and third phosphates. All right, we've got an adenine nitrogenous base. We have a 5-carbon sugar, or ribose, and the 2 phosphates with this third one that pops on and off, releasing energy when it pops off, and then storing energy in this bond right here when it is added in. An analogy for this is the stored energy in the compressed spring of this pop gun. Then can be that when that energy is released, but the potential energy stored in the spring is released as kinetic energy, that makes that little dart zip forward and stick on someone's forehead. Cells use ATP for many different things, including mechanical work. Think of the flagella moving on the trichonympha that we were looking at. They had many smaller flagella, but this organism has longer ones, so the actual motion. Cells also use energy for transport work, which we talked a little bit about last week. Active transport, moving something from an area of low concentration to high concentration. That's backwards from what diffusion wants to be doing, right? So we need to use energy to pump against the concentration gradient. And then also in chemical work, although enzymes greatly reduce the amount of energy that a reaction needs in order to happen, they do often need some ATP. So here's an example of an enzyme working with its substrates and products using ATP to finalize that synthesis. So here's another one of the same reaction. We have adenosine diphosphate plus this lonesome phosphate molecule. Energy from the breakdown of food in any organism is stored then in that power bond here. We can consider this, so that's ATP, adenosine triphosphate, which is a high energy currency that can be used in any cellular reaction around the cell. Okay. Here is a diagram with the reaction for cellular respiration, also known as aerobic respiration, because it involves oxygen. Using the 6-carbon sugar glucose in the presence of oxygen, cells can break that down into carbon dioxide and water and ATP energy. Depending on who you're talking to, it's between 36 and 38 ATP produced. My general gist of this is that aerobic respiration makes quite a bit of ATP for each molecule of glucose used. Right? And key and important thing here is that we've got oxygen involved. Okay? Cellular respiration is the reason that we need to have a respiratory system that transfers in oxygen into our cells so that they can do cellular respiration and then get the carbon dioxide out. Okay? So because our cells are doing cellular respiration or aerobic respiration, we need to have a way of getting oxygen in and getting carbon dioxide out. So that's our respiratory system. And that can, if you can't remember which way the equation goes for cellular respiration, just think about breathing. Okay, you're breathing in the oxygen, so that's got to be a reactant, and breathing out carbon dioxide, so that's a product. This is all going on in a mitochondrion. Here is a micrograph of one showing the outer and inner membranes, the matrix, which is the cytoplasm of the mitochondrion itself, has its own name. All right, and there's also an intermembrane space in between the outer membrane and the inner membrane, just to make it really exciting. Lots of surface area here. All right, here's a artist's rendering of that a little bit more simply. Same shapes and spaces. So breakdown of glucose begins outside of the mitochondrion. All right, it goes through glycolysis, and then the pyruvate is brought into the inner membrane space to begin. And then we're going to end up with a concentration gradient across these between the inner membrane space and the matrix within. I would assume that your lecture professor will go into this in much more detail. What I want you to know is that this is happening in the mitochondrion, and we're producing a lot of ATP when oxygen is available to complete the process. This is the image from your book. I'm sorry it's so blurry. I was trying to make it bigger, but obviously, that's painful. But look at it in your book. It's better. This is a really nice breakdown of what's happening where, and pretty simple. Citric acid cycle, what's coming in and out, right? And getting into the electron transport and oxidative phosphorylation, which is a really fun word to say 8 times fast. I won't do it right now for you, but you try it. Okay. What happens, though, when there isn't enough oxygen? All right, you have been running away from this tiger as fast as you can, and you're almost to the tree, but you're really running out of oxygen. Your lungs can't get enough in fast enough. Luckily, our cells can do anaerobic respiration, also known as fermentation, in order to produce just a little bit more ATP to get those muscle cells going a little further, to get up the tree, get on the bus, whatever it is that you're running for. Okay, so in human muscle cells and other animals, all right, glucose goes through glycolysis to produce the pyruvate, but because there isn't any oxygen available, the electron transport chain can't happen, and we can't do the full cellular respiration. So the pyruvate is converted to lactate, or lactic acid, right, which does the whole NADH regeneration thing, which I'm not going to ask you a whole lot more about, but just so that you have it in your head. And we get a whole whopping 2 ATP left. Okay. But that's good. That's a little bit, right? That little bit of ATP that's produced might get you, like I said before, up the tree or on the bus or whatever it is that you're running to, needing that little bit of extra energy. If you go in the slideshow, if you're interested, there's a great Scientific American article looking at the evolutionary adaptation of this lactic acid and pathway, and why does lactic acid build up in our muscles when we're running, and that causes that initial burn, right? You know, your muscles are burning in that last sprint, but it isn't what causes muscle pain later on. So it's a feedback mechanism. Eventually, it gets so acidic in your muscles that that cellular respiration can't happen. The muscle firing can't happen. So you stop. Otherwise, you just keep going. But check out that article if you're interested. Applying this stuff in real life. Okay, so that's human and animal cells. Another kind of fermentation happens in yeast cells and other microorganisms, where instead of producing lactic acid or lactate, they are producing ethanol, all right, and the carbon dioxide. So they do the same glycolysis, pyruvate, but that's converted to ethanol instead of lactic acid. Okay, and this is true fermentation, and produces all sorts of interesting things, which I'll talk about in a second, but also that 2 ATP. All right, so not a lot of ATP, but some, and they don't need any oxygen to do it. There's those budding yeast cells we looked at in the cell lab. So fermentation is anaerobic, which means no oxygen, but carbon dioxide is still produced. Right? And there are a lot of commercial products that make use of this amazing process. So our friendly neighborhood glass of beer here, right? It's got the ethanol, that's alcohol. That's why people like to drink beer. It also has the carbon dioxide, those bubbles, that foam that you poured so carefully. That's the carbon dioxide that was produced in the fermentation. Okay. Bread also uses yeast fermentation, right? That's the thing most people think about when they think about yeast, right? But why don't you get drunk off your peanut butter and jelly sandwich? What happens, right? We still have the carbon dioxide that's produced is what makes those lovely fluffy little bits in your bread, the air bubbles. Okay, it gets caught in the gluten of the elasticy gluten of the wheat protein, and as it's rising, that's what makes the bread dough rise. But then when we bake it, all the ethanol that's produced bakes off. So there's actually not any alcohol left in bread. Same thing when you're cooking with wine, or if you're sauteing, you cook off the wine, and then you're not drinking it to become inebriated or something like that. Not that any of you would do that, but at any rate. Okay, so please read through the lab. I strongly recommend that you read carefully through the introduction to Lab 7. It has a lot more information on cellular respiration and fermentation. And then check out the exercises. Please write your hypotheses on page 80 for looking at the germinating versus dry peas and boiled peas versus germinating peas. And then on the fermentation section, which is exercise 2, there's another spot to write a hypothesis. That's on page 84 if you have page numbers in your manual. Thanks. See you in lab.